translational approaches in plant system is a very apt topic for this session, because ultimately we need to understand that today's knowledge leads to tomorrow's technology. And when there is a technology which should result in translation, that means the new product should come out of it, new innovation should come out of it, and that is the reason I have kept the title of my talk as Innovations in the Area of Agri-Food, Nutrition, and Biomanufacturing. So in next 30 minutes or so, I'm going to give you a brief glimpse of what we are doing at the institution when it comes to handling the plant biology in the form of translational research. I have used the word biomanufacturing over here for the benefit of students. I'm sure many of the students will be attending this talk. Biomanufacturing is actually a very beautiful word which encompasses all the processes associated with the innovations and the technologies which you have, where you can get a translational product out of the processes which are happening otherwise in the nature. You modify those reactions, you change those reactions, you speed up those reactions to do something which was otherwise not possible. So in short, we can say that biomanufacturing is nothing but putting nature to work for the benefit of mankind. This kind of biomanufacturing is very, very important because, as all of us know, that the world population is increasing every day. We are going to be 10 billion very soon, and I think all the students should know that the population of India is already the highest in the world. And I also want to tell that under these circumstances, when the population is increasing, the agricultural land which is available for agriculture is decreasing every day because of urbanization, and the crop productivity which we witnessed in the form of green revolution a couple of years ago is at stake at the moment because of the climate changes. The monsoons are becoming erratic, salinity, drought, high temperature, low temperature, flood, insects, nematodes, viruses in the form of both biotic and abiotic stresses are actually resulting in decline in crop yield. And for us to be able to increase the income of the farmers, we have to perform innovation or translation ideas when we can get best out of the waste which is generated in the agricultural form. So this talk is basically going to talk about innovation and the experiment and the technologies which we have developed at the two institutions at the Department of Biotechnology. I'll just keep on giving you brief ideas, and in case any of the students has interest to join our institute for research or for postdoc, you're most welcome. You can contact me later on. The product which I'm going to talk about in this slide talks about golden banana. We call it the next generation superfood. While you can see the traditional banana, which is shown on the left side, which is white in color, all of us eat it every day, we have developed a banana by doing genetic modification, which we call golden banana. And you can see the golden banana as it is shown over here on the right side. It is not only golden in color, but it's golden in qualities as well. Many of you would have heard about golden rice, which is rich in provitamin A. We have gone a step beyond it, that we have not only made vitamin A available over here, but iron and disease resistance has also been incorporated in this golden banana. The initial technology, which came from University of Queensland, QUT, has been modified, and we have now gone at the stage when we have not genetically modified banana, which is giving the vitamin A requirement as per the RDA, required daily allowance, but we have also generated through gene editing. And Government of India has already given nod for consuming or growing the product which comes from genome editing or gene editing, especially from SDN1, sided nuclease 1 and 2, which do not leave any foreign DNA into that. So we are very excited about it, and we believe that we have reached a stage when we can claim that even half of the banana is good enough for the children to take care of vitamin A deficiency. So this is something which is going to change the whole way we talk about the diseases. And this is an example when we can say that the plant science has reached a translational stage and the product is ready for the biofortification of banana. Another product which we have developed at Navi is another colored product. The previous one was golden in color. This is black or purple in color. And in this case, it is not banana. It is a staple food. Yes, we are talking about wheat over here. As you can see, different colored wheat over here. This is because of the presence of different anthocyanins that develop naturally in the field at the time of grain filling. So we have several products from these colored wheat, amber, black, blue, purple, and we have already reached at the stage when these kind of products are available across the shelf in many areas in Punjab, UP and Haryana and Madhya Pradesh. Almost 24 progressive farmers are already cultivating these kind of black purple wheat which we have developed at Nabi, and they have also been the program of government Poshan program at in Punjab area. And these kind of food are very, very important because they help the human beings when we eat them for fighting the diseases, because the anthocyanin, because of which the color is different, they are naturally occurring antioxidants that gives goodness in fruit and blueberries which we buy at very high cost. And since we had developed in wheat, which is the food for the poorest and food for the richest, we believe that these kind of interventions are going to play a very, very important role and ensure the food security in terms of the product development. Another beautiful technology which we feel happy about is related to improving the shelf life of the fruits. Generally, you must have seen, farmers, they harvest the seed grains out of the seed of wheat, and the husk is left behind, which is often used as a waste, or sometimes it is also used as a food for animals as well. So we thought of giving value addition to this wheat straw, and some of the chemists at our institution have developed a wonderful formulation which is totally vegetarian in its nature, and we have tested it on a range of fruit including banana, pear, apple, orange, guava, and what we have seen is that the formulation which we have developed based on the agri-food byproduct, which include wheat straw, oat bran and potato peel, is able to improve the shelf life without refrigeration of the fruit for up to 30 to 40 days, and peaches as long as 6 to 8 days. This is going to play a very, very important role, because all of us know that food is, when it is coming to metropolitan cities such as Bangalore, Delhi or Mumbai, we do not have agricultural land close by. The fruits and vegetables are actually flown in, or they are brought in from the neighboring villages and the state, and almost it is believed that 30 to 40% of the food or the fruits which we are getting are lost because of the post-harvest losses, because when we transfer them from the site of production to the site of consumption, there is already a loss of 30 to 40%, which is a huge number. So using this technology, we have tested it on several range of fruits, and we have tested it in Punjab in many cold storage areas as well, where it has increased the shelf life of the apples for up to 6 months. We believe that this kind of product are going to play a very important role, because these are totally vegetarian in plant-based product in the nature, as compared to many other products which are shellac-based, which is animal-based coating. And since these are plant-based products, even if you consume it, they are safe for human consumption. And of course, they are cost-effective, simple, and can increase the income of the farmer beyond the income which he or she is generating by selling the seeds out of it. We also try to make use of other waste which is generated under field condition. Shown over here is an example where we have developed nanocellulose-based biofilm, which is being used for many applications. Shown over here is a biofilm which we have generated on the left side. You can see that the fruits which are covered by the biofilm, which is coming from the nanocellulose, which we have generated again from the waste which is generated in the agriculture field, is able to improve the shelf life and the quality of the fruit which is shown at the bottom of it. And it is a huge market potential, and these technologies are patented from our institution. We also are trying to develop solution, and this is a prototype shown over here, which we have made with the help of CSIO, CSIR. Shown over here is a mobile-based device which we call Bactosensor. It is a smartphone-based biosensor for bacterial detection. You must have heard, children, that the food which is left untaken, which is not taken care of, which is not put in the refrigerator, it gets spoiled very easily and cause food poisoning and stomach upset in many of us during summer season. This primarily happens because of the food spoilage by the bacteria. The common three types of bacteria which we have finalized. We have this mobile-based sensor which can detect the presence of these bacteria. It's a special technology which is aptamer-based, which can detect the presence of the bacteria within, say, a few minutes. So this is the kind of, a point-of-care aptamer-assisted multiplex smartphone. We call it multiplex because it can detect the 3 bacteria simultaneously, and it can give you the color which is shown in the form of dot over here. So even a person who is not so much trained in handling the molecular biology, he or she can actually detect the presence of bacteria in the food and hence can prevent food poisoning and prevent many of the people from falling ill. So this is the kind of very affordable and simple to use bacteria detection. It can be used to enhance the food security at national level. It may have a lot of application in the catering industry and flight and aviation industries as well. Our center is trying to develop new product from the lignocellulosic biomass. I just showed you one biofilm, but besides biofilm, we have other cellulose derivatives, which is coming from the rice straw or the wheat straw, which otherwise is a nuisance. The farmers, they burn it and causes a lot of pollution in the month of November and December. We have the technology, which you put in the technology, we can convert it into wound dressing or hydrogel. We can convert it into coating and also bio-based chemical, which includes different functional biomolecules. I'm going to give you a flavor of these things in a minute from now. Shown over here is a slide that talks about agriculture and horticulture waste. We have been able to convert it to cellulose derivatives from rice straw, wheat straw, apple pomace, and of course bluegrass. The nanocellulose which we generate can have, as I said, several applications. For example, for hydrogel, which is used for drug delivery, for wound dressing as a kind of membrane, and of course we can also make a film out of it, biofilm, for packaging, which is totally biodegradable and not as plastic, which remain in the nature. So these kind of conversion of waste to the best technology is available at our institution, and they have been patented. Let me tell you that some of these technologies are at the testing stage at the industrial level, so we are very hopeful that these things will see the day of light market very soon. This slide talks about pectin nanocellulose-based hydrogel for drug delivery application. These are basic chemistry, which we will not go into detail of it. But what I want to show from this particular slide that they can be very useful for curable infection diseases, because these kind of hydrogels are very important because they are biocompatible, they are biodegradable, they are porous, they are stimuli responsive, and exist in various forms. And these kind of innovations have huge market potential, as can be seen, and we have filed a patent for this as well. We also have technologies which can convert it into chitosan, gelatin, or these kind of membranes, which are very important for wound dressing. The important thing about this wound dressing will be that they are compatible with all the human beings, and they will not cause a skin allergen, and of course these are biodegradable as well. And it has a lot of potential as a market value, which can be seen from the figure which is there on the bottom right of the slide. Another interesting area from our food technology group is towards understanding the browning of white adipose tissue which causes obesity. Earlier we used to believe that Indian population is not so obese. It's only in the Western culture the obesity is there. But I think slowly we are realizing that Indian children are also being facing the issue of obesity, especially the abdomen obesity is happening very easily. What we are trying to investigate in this work is the role of the traditional food in the form of ginger, cinnamon, mustard, chili, mint, and garlic and we are trying to understand the components which are present in these food, which does not allow conversion of white fat into the adipose, white into the brown adipose tissues. This is what we are trying to do, and we are trying to develop some biotherapeutics out of it. We are also working towards finding the efficacy of symbiotic, which include both probiotic and prebiotics, based on the traditional food. And in this case, we are talking about prevention of obesity, diabetes, and several other diseases which are immune related by using this formulation, which are primarily coming from the traditional food, such as millets and all that. Lignin, which is often referred to as brown gold, it is believed that it will change the market as far as biomaterial market is concerned, because it has many uses associated with that. Some of these uses, I will not go into detail of that, is listed in the slide. It is second most natural biopolymer, available in plenty, especially where the agriculture is being done. It is biocompatible, it is adhesive, it is antimicrobial, UV resistant, and the lignin-based nanomaterial, they are widely used as antioxidants, antimicrobial agents, and UV resistive coatings, such as drug delivery vehicles are also being generated. What is shown over here in this slide is lignin-based photoactive coating for antimicrobial applications. We have developed lignin-based formulation, patent has been granted on that, where we are able to convert this lignin nanoparticle for the purpose of bioimaging, bacterial inactivation, and as UV protective agent. So they have, again, huge market potential, and these technologies, some of them have been transferred to industry for testing as well. Again, the lignin-based hydrogels have been used for drug delivery applications, because they are very, very useful for drug delivery platform, and they also are being used as antimicrobial agent, and they are wonderful adsorbent material, again having market potential for that. We also have a center of excellence in lignin over here, where we are developing several products, which is coming out from the rice straw, which otherwise is a nuisance for the pollution. Shown over here is an application where we are trying to generate lignin-based antimicrobial sunscreen and cosmeceuticals. Several of the products have been generated, they have been tested, and the beauty of these kind of products is that when you actually soak them with zinc nanoparticle, the color of the screen is matching with the skin color, which is shown on the leftmost side. While the cream which we use, which is available in the market, they are white in color, so people don't like it. So if you have these kind of products which are being used for cosmetics and UV protecting agent and antimicrobial agent, again they have a lot of market potential as far as translational product generation is concerned. Shown over here is the way by which we can convert lignin into PHPA, which is then used for cosmetics. There's a lot of chemistry involved in that. Let us not go into that, but again, this polyhydroxybenzoic acid, PHPA, has a lot of chemical value beyond cosmetics as well. I will not go into detail on that. Another area of research which we are very happy to mention is that we are trying to develop solutions for diabetes in the form of zero-calorie sugar or low-calorie sugar. So what we have done is, we have technology by which we can convert the D-fructose into special kind of sugar, which are almost zero-calorie sugar. And for this, we have engineered the cell with specific enzymes which are thermostable, pH stable, and they have been patented for their qualities as well. And we are happy to say that the D-allulose which we generate, which is the zero-calorie sugar, it has 98% purity at the gram scale. So these kind of products which we are again generating out of the waste which is generated in the form of the sugar cane and all that, and also by using the fructose available in the market, they are anti-diabetic, they are anti-obesity product and anti-dyslipidemic as well, which are taking care of a lot of lifestyle related diseases, health problems in the modern world. We have also technology by which we can convert the cellulose into byproduct, And shown over here are oligosaccharide. Efficient process engineering has been developed, which is in the form of integrated, in the form of corn fiber. And we can use it for the extraction of maltose, hemicellulose, and cellulose, for which we have the patent, having applications in pharmaceutical, cosmetics, and food as well. Some more byproduct which we have developed, the quinopomace, which is again a waste in this area, in Punjab area. We have technology by which we can do debittering, and we can take out the edible fiber and use it for various kind of product in the form of vermicelli and all that. We are also producing a lot of good industrial product from the rice straw. As I mentioned, rice stubble is burned after the farmers are able to take out the seed, harvest the seed, which causes pollution. So a lot of technologies which are not only solving the problem of pollution, but also giving good product in the form of hemicellulose and cellulose, which can convert into sugar alcohol, and pigment, which are having applications as antioxidant agent, low calorie sweeteners, and they are also known to prevent inflammations as well. We are talking about xylitol over here. We can also convert it to prodigiosin, which is again antioxidant, anti-cancerous, antimicrobial, anti-malaria, and we have got patent onto that. I'm not going to detail of it. I'm just trying to excite you, by which we can understand the kind of diversity of work which is being done at the institution, giving extra income to the farmers and also solving the problems which are associated in today's world related to the pollution, which are related to stubble burning in this region and many other states in the country. We also have a very quick method by which we can convert the flowers which are otherwise based in some region, like this technology is talking about the flowers of Kalash, Bugia, monosperma. We can convert them into natural dye, which is totally non-toxic and edible. And we have a simple process, one-step process, which we can generate out of it. We have license to several companies over here. We have technologies by which you can generate blue dye, red color dye, which is water soluble, oil soluble, and brown dye. So these technologies have already been transferred to the leather industry and textile industry, which are, these are natural dyes you're talking about, which is giving the new inputs as far as the waste which is generated by these processes. We're also producing biofertilizers and biopesticides from agri residues. I will not go into detail on that. Again, this technology has been patented and transferred to an industry. We are also producing the novel solutions such as citronella oil. We are able to produce it for the pesticide spray and the perfume which we are generating out of it, which are very, very popular, and they can be used as a fabric roll-on, mosquito repellent lotion, and these are all coming from natural products, so there is no toxicity associated with that, and we have the patent on these PMDs and all that. So this is the slide which talks about other products which are we are working towards it, which are at different stages of TRL level. We talk about enhanced shelf life, which is non-toxic, facilitated storage. We have foodborne sensors for food diseases. We have lignin valorization. We have black colored and different colors of wheat, and we have nanofilm and tissue engineering. So with this, I would like to stop over here, giving you an idea that what kind of different activities is being done. And I also want to tell you that this institution is one of the premier research institutions situated in Mohali. We have several interesting facilities which are available for research in the form of, say, supercomputer. We have Param Smriti supercomputer, which was inaugurated last year. It has a capacity of 670 teraflop, and we have a national speed reading facility also coming up within, say, 10 days. For the benefit of students, I can tell you that in the farmers field, the farmers can get 1 or 2 crop in certain areas, especially in southern areas, say for rice, they can take 2 crops in certain part of the area, but in Punjab they can take only 1 crop, such as rice, in one season. But the availability of speed breeding facilities, which are very innovative technology, disruptive technologies, I would say, allows us to take more than 1 crop, sometime up to 3 or 4 crops within a year. So it is going to not only give you the next generation when you're doing experiment, you want to test the effect of something on your 4th generation, so you can do that in 1 year. So it is going to speed up the research pace by which the scientists are doing research, but it will also help in generating new varieties at a faster rate. And this together with the Param Smriti. And I also want to tell you that we have also been recognized as a National Center for Training in Gene Editing, which is coming up in a revolutionary way for the development of product, which were otherwise not possible by classical methods. So a lot of opportunities exist at the two institutions, a lot of good infrastructure. So I would like to welcome one and all to visit our institution. And my sincere apologies that I just could not join you personally, because this facility is at the last stage and we are at the process when we are going to inaugurate, and a lot of work is related pending to that facility. So with this word, thank you very much, Professor Hari Majumdar and organizers at SNU for giving me this opportunity. And I'll be very happy to take questions in case any of the student has it. Thank you very much and wish you all the best.
It's a pity that we couldn't have him in the person, but I'm sure that at least for the students who have been listening carefully, if you have any question, please put them down and they can be collected and perhaps passed on to him so that he can get you back with the answers. So with such an excellent start to the last session of the day, our next speaker is Dr. Sucheta Tripathi. She is the Senior Principal Scientist at the Structural Biology and Bioinformatics Division of IICB. She is Fellow of the West Bengal Academy of Science and Technology and member of the Advisory Committee, Research Advisory Committee at NTRF, National Tea Research Foundation. She is an invited chair of China UMI Site Molecular Genetics Network in Nanyang University. Her research, she is a Ramalinga Swamy Re-Entry Fellowship of the DBT and also invited chair of Omnian SLU, Sweden UMI Site Molecular Genetics Network. Her research work is in the area of dissect the genomes of the indigenous microbes, namely cyanobacteria and fungi, and understand their genomics using second and third generation sequencing technologies. She is also trained in use of rapid RNA sequence technology to understand the transcriptional changes. She has engineered the genes for commercial production of biometabolites and is familiar and user of big data analysis using their own in-house software. Dr. Tripathi.
Professor Thakur for the nice introduction, and also the organizer, especially Professor Lahiri Majumdar, for inviting me here. And this is one such plant biology conference with a genomics in it, and that's why I'm very excited. And I will tell you my story, which is not going in the correct way that everybody does. They have a hypothesis, they do experiments, and they go to the genomics. I go the other way around. I come from genomics and then do the other things. So that's what I'll take you in a ride. As Professor Thakur has already introduced, that I am interested in Indian microbes, especially cyanobacteria and pathogens. Before joining IICB, I was working with O. mycetes pathogen, which is a pathogen and which is not a fungi. And we did a lot of work, but after coming back here, I wanted to do something else. And before I start with my acknowledgements, because these are the people whose work actually I am presenting. So of all the people, everybody has contributed, but I will be talking about work of Dr. Bonita Das, Asha Rani Prusti, Shubhajit Datta, Geeta, and also Dr. Mayuri Mukherjee and Dr. Diksha Singh, who has contributed to this work, and also my funding agencies and my collaborators and everybody else. I am not taking time talking about each of them. So one of my collaborators is here, Dr. Smriti. So why cyanobacteria? Cyanobacteria, I know that, when we are talking about plant, we know plant is coming from cyanobacteria, right? The cyanobacteria became the plastid, and eventually it has made a heterotrophic cell into a phototrophic cell. And why we are interested is, if you look at the evolutionary history of Earth is about 4 billion years old. And this organism has almost coexisted, I would say that its history of 2.5 billion years, and it has sustained itself. And that is what the remarkable resilience these organisms are having. And since these are prokaryotes, we are very intrigued to understand what they must be doing and how they are doing things. And we know that there are cyanobacterial blooms, and when the blooms are happening in the ocean and the rivers, you will see the aquatic life are getting affected. So they are producing toxins, and these toxins are actually nothing but they can be also used as drugs, and that is why we are interested in studying them further. So I will be just showing you few cyanobacteria we have collected from different parts of the country, and these are indigenous cyanobacteria, and I am sorry to say that we do not have a proper repository where you can just go and buy this. We have collected many from the desiccated environment, and some I have borrowed from my professor, and some of them, these are the hot spring organisms, and this is isolated from Lakshadweep, it's from the ocean. And this is also oceanic cyanobacteria, and of course we have many more. I am not showing you that, but I will be talking about tales of these 3 organisms, which can, from the genomics, how we are trying to, we have not translated yet, but we would definitely like to do something which will be useful for the human health. So as I said that we always use a top down approach, so we get to the genome first. And after we have the genome, there is a whole lot of science there, that is the science of bioinformatics, where you assemble the genomes and then you find where the genes are. And that itself is a very complicated process. And after we do that, we try to understand how these genes are expressing doing a bulk RNA-seq, and thereafter we have our hypothesis. So everybody says that give the DNA to the company will do it, and company definitely does it. But it is not very easy. I will show you why it is, and this is work of Asha. I will show you this video. Okay, here I pause it. See this organism gliding over this. This is called as a ciliate. If you have a culture of cyanobacteria, I think you would have heard about Nivedita's lecture yesterday, that many of the organisms, as we think that they are occurring alone, that is far from truth. So they co-occur together, and there are ciliates and many other organisms, we cannot separate them, and they cause lot of problems when you are doing a genomics or analyzing the data. So therefore, I am just showing you some of the pictures of this is one organism called as, this is a new species, novel species, and we have identified, and this paper is almost through. So I will talk about this also. So this one is called as Oscillatoria salina, which is also isolated from the ocean, and if you see that it has remarkable pH bioremediation properties, that if you put them in the acidic environment, immediately it turns into alkaline. And this is the Masticocladus laminosus, which is a hot spring species that also I am going to talk about. You just look at these cells, how polymorphic they are. Some are, although it is prokaryote, you can see that some of the cells are dividing in a vertical manner, some are dividing in a horizontal manner, and they belong to the most advanced category. And these are the differentiated cells, which are called as heterocyst, and heterocyst story is altogether separate science, because it is the specialized cell which does nitrogen fixation, and there are many genes regulated in this. So how we are using genomes for understanding certain phenomenon. So I will tell you, we have used genomics as a tool to understand, first of all, to correct the taxonomy of many of the cyanobacteria. So in this process, actually, I was checking yesterday, we have submitted 97 genomes to the GenBank. I think the single largest lab so far from India. So if you look at the pipelines, you see when you are trying to get the data and you want to use some of the methods, you see that each of the methods, each of the steps you are doing, there are multiple methods available. So if you choose a particular method, then the results will be completely different. So therefore, you have to optimize your pipelines so that you get to the most optimized assembly from where you can do the downstream data analysis. So I can show you one example. We have one species that I said is a novel species which is almost there. So we have named after our institute. So this organism is there with us for the last 10 years and we have sequenced it 3 times. And you see the first time when the methods were not very advanced, we got 16 fragments. We can call it as 16 scaffolds. Then we did again in 2014 we got 3 scaffolds and now we got 1 single chromosome. So this is the advancement of the pipelines etc., which gives us better resolution of the genome that we can use it for the downstream analysis. So here is one of my very talented students, Nivedita. He is from also Calcutta University. He works very, very hard. So he has done high throughput genomic analysis of this organism and all the existing cyanobacteria, high quality cyanobacteria from GenBank, and he showed that this organism is actually a novel species. How? We have submitted a genome announcement when we sequenced for the first time, and based on the morphology we call it as halomicronema. And thereafter another group published a paper and they thought that their genome is very similar to us, so they named their organism as also halomicronema. So now after we do everything, we find that our organism is not halomicronema. So we are pursuing them to rename their organism so that it belongs to the genus that it should be. We call it as Leptolyngbia iicibica, and it was through this extensive amount of genomics and 16S analysis that Subhajit has done, we have come to a conclusion. And there is also another student, Gita, who is also working with a set of cyanobacteria, hot spring cyanobacteria. It belongs to a family called as Hapalosiphonaceae, and Hapalosiphonaceae family is a very heterogeneous family. It contains lots of different diverse habitats of cultures. So it is very difficult to put all of them together as one family. So she also carried out extensive genome analysis and she came up with a conclusion that Hapalosiphonaceae has to be splitted into 3 parts. One will be which is into a very localized habitat where it is not undergoing evolution. Another one is which is rapidly evolving, and the third one is a mixture of heterogeneous group. So these are all unpublished work, but at a various stages of revision. So this is also one of our contribution using genomics. We are proposing that the taxon names have to be corrected in GenBank. So also again, as I said, we use genomics heavily for everything. So we wanted to also understand how the organisms are undergoing or tolerating the stress mechanisms and their resilience against these stresses. So again, Dr. Mayuri Mukherjee, who has completed her PhD, and Ms. Geeta, they did some pilot experiments, and they found that these organisms, when they are growing under culture, something very remarkable happens on the 3rd day, 9th day and the 12th day of their culture after the media has been supplemented. So we have taken these as the time points and we did extensive transcriptomic analysis, and our take home message is actually very interesting. And the message is, like, we know that in human cases also, when we don't do physical activity, what happens? Our muscles become very weak, and if we don't do brain activity also, the brain becomes very weak. Same thing happens with this organism, cyanobacteria, because this Masticocladus is actually it can fix nitrogen. Okay, so if you don't give external nitrogen source, it can fix from the atmospheric nitrogen. When we grow them with nitrogen source and without nitrogen source, we found that when you are giving nitrogen, this organism is losing fitness. And that was found with multiple side effects, like it has lost its ability to move, it has lost the ability to regulate its photosynthetic mechanism, and many side effects were there. And we also, for the first time, we sent this paper to Plant Physiology, and we said that in presence of nitrogen, these organisms are going to have a sporulation. So the paper revision came back saying that in cyanobacteria, there is no report of having any sporulation. So that was the only thing they said, and they rejected our paper. So subsequently, what Geeta did, Geeta showed that these organisms do have sporulation. Not only they sporulate, they also go into dormancy and they regerminate back into normal stage. So we have proposed a detailed mechanism of the action, what happens when the nitrogen is given versus when the nitrogen is not given. When the nitrogen is not given, the organism sporulates. That means it tries to sustain itself and it tries to protect itself. But in presence of nitrogen, it has a lot of side effects, that is, it has DNA damage, it cannot do photosynthesis, it becomes a complete mess. So this is also another schema to show that what we have come out of this genome and transcriptome analysis, that how this is affecting the motility of the genes and the detailed mechanism of this organism. And from that, so these are the spores that I said Geeta has produced, and they are germinating, you can see, with a terminal heterocyst. And this is a video to show whether it will form, I don't know. So also we have found from our data that when you look at the morphology of this organism, heterocyst, as I said, is a very specialized structure, and if you look the type species, which is nostoc, you will find that heterocysts are organized in a pattern, that one heterocyst is separated from another heterocyst by a given number of cells, like 10 or something always. So there is a pattern always, and the gene which is responsible for the pattern formation, we found that this pattern formation gene is mutated in our organism, and therefore you can see that there is chaotic heterocyst formation. You can see here, there is no such pattern. So the detailed mechanism of heterocyst delineation and heterocyst formation has been discussed using the genomics and the transcriptomics data. So this is our take home message. This is kind of a summary that we can say that when we go to the rice field or somewhere, you see that people are giving lot of nitrogenous fertilizers, and nitrogenous fertilizers could be detrimental for the cyanobacteria which is growing. So we have a cousin of Mastigocladus which is called as Vestilopsis, and it is very similar to that is found in the rice fields. So if you give lot of nitrogenous fertilizers, these organisms eventually will lose their fitness. So therefore the ability of the organisms to have a symbiotic relationship or to make the organism, to make the higher plant productive, will go down. So another part I would like to talk about, that is how we are going to exploit from genomics, that how we can use it for our human disease problem, solving human disease problem. So the organism Oscillatoria salina that we have used, we have again done extensive genomics, and Bonita and Asha both work together, and in this organism, while analyzing the data, we found a lot of metabolic gene clusters, and these metabolic gene clusters were identified as having a lot of potential drug like ability. Similarly, the Mastigocladus I showed you before, it has something called as chinorin. Chinorin can be actively used for sunscreen type molecule. So from here we decided to go further and test the organism, and there you go, we have isolated compounds, and we have seen that it is having anti-spheroid activities, that is from the hot spring species, and these are the differences are quite significant actually. And in all the three species I am talking to you about have anti-cancerous property, and this is for the first time we are showing it, and this paper is not published, this is under review, and we have done all kinds of assays to show that the water extract of this organism is actually more viable than the methanol chloroform extract. So I will skim through these slides quickly. And also we have shown that these are antimicrobial activities against a plethora of pathogen, very deadly pathogens. So from here we would like to take it forward, and we would like to take it for large scale production, which is where we are standing. So this is all about from genomics to the translation, I would say. Thank you for listening to my talk.
This paper is open for discussion. Any questions?
When you are doing this genomics and renaming and new genus, new species you are naming, so which classification are you following?
There is a standard method when we are doing with respect to the genomics distance between the organisms. So if the genomes are at least 95% apart from each other.
No, that is known, but you know this molecular classification, the Comarex classification, Comarex is going to publish lots of books.
Right.
And for cyanobacterial identification, only genomics won't.
Yes, the morphological classifications are also there. So we are checking we are actually not very strong in that area. So we have not done it.
No, it is my advice, working with last 40 years with cyanobacteria. You first you go for morphological identification. Follow Deshikachari's book.
Okay.
It is very basic and they have given everything. And if there is any confusion, then you go for genomics. And then you have to consult Comarex. Books are there, monographs are there. Okay.
But these organisms are not listed in those books.
Which one? Mastigocladus?
This Halomicronema,
No, Halomicronema.
Leptolimbia.
Leptolimbia is already there in Comarex classification.
Halomicronema,
I don't know whether it is Mastigocladus. That's why I am telling it may not be. Then you can go for new genus, but you first consult all these books.
Yes, first what we did is we did a morphological classification based on their length, their width and their...
yeah, it's all right. I am giving the suggestion just to consult.
Sure.
Deshikachari and many others books are there. Okay. Then you go for genomics.
Sure.
Okay. And for this combination of morphology and molecular, 16S or I don't know what you are doing, what marker you are doing. You have to go for Comarex books. Consult those.
Okay.
Then, because people are making new genus every day. So it is confusing, much more confusion in cyanobacterial classification.
Actually, it is polyphasic, so many things are there, so they may appear very
close.
No,
you have to refer all these Comarex classification because there they are naming, so it is different, so you can go for new genus, no problem in that. You can go, but you consult.
Sure.
Okay?
Thank you.
That is my suggestion.
Sucheta, what is your interest in cyanobacteria? Let me complete it. Is it the genomics of cyanobacteria or biomimetic analysis or classification of the existing cyanobacterial group?
Actually, I started off with this metabolic re-engineering is one of my most interest area where I want to discover the metabolic gene clusters and I want to see that whether I can put them into some other vector and clone them or overproduce these metabolites.
Does it require a lot of classification studies?
No, it doesn't require. While we worked, we found this was very interesting for us to see that this was actually novel, this organism.
My experience with the cyanobacterial system is this, that if you want to do this genomics part of it, bioinformatic analysis of the genome of the cyanobacteria, and your interest is the metabolic cycles or metabolites. I also saw that there is a metabolite search. You really don't have to worry about the classification of this, and don't spend your time on the classification and relationship of this. These will be done by taxonomists, cyanobacterial taxonomists. When there is a confusion of classification of one particular genus, assigning them to a particular genus or species, then only you need to do the genomic study and to identify the strains or the genus or whatever. Since your interest is not in the classification or the taxonomy of the cyanobacteria, you better stick to whatever you are doing. It's my advice.
Sure.
I do not have 40 years of experience in cyanobacteria, but some experience in cyanobacterial genomic studies as well. And you probably know that. And the other questions that, this is only a suggestion and you can think about it. Talk to your director or whoever your RAC members. And the second part is this, is association of the cyanobacteria with a particular group of plants for nitrogen fixation. I wish you were there yesterday when Anindita Sheel gave her talk.
I worked with Anindita. We worked together.
I know, I'm aware of it.
Yeah.
But if you had been here yesterday.
Yes, I heard a talk before also, SBC and
yesterday there was a lot of discussion on this. My question pinpointingly is this: if you starve a plant of nitrogen, with soil and nitrogen, and can you induce by some kind of association, cyanobacterial association, the ability in a plant, the nitrogen fixing or the nitrogen assimilation pathways? This is something which has not been done. But her work tells us that there are some microbial associations which will probably do it. And the indication that you gave in your talk about this may be continued. This is my suggestion to you.
Sure.
And I won't be able to do this kind of work, but you can do that.
Sure.
Please bear this in mind. Okay?
Sure.
And genomic studies of cyanobacteria is a very important part. I suggest that you continue with that work.
Thank you.
Thank you.
Thank you, ma'am. Thank you for the great presentation. And I just wanted to know that you have talked about the cyanobacterial metabolites. So are there any metabolites commercially in use for making some drugs? For you have told that it is anti-cancerous. You have found that it is anti-cancerous. So I just wanted to know.
I think they have found the mechanism, but to make it commercially viable, they have to overproduce. So that is the bottleneck right now, to have them produce in large quantities so that it can be cost effective for commercialization. But otherwise it has excellent properties for, in terms of anti-cancerous properties, antimicrobial properties. I am not aware of any compound which is right now derived from cyanobacteria or in market right now. That I do not know of.
Okay. Okay, ma'am. Thank you so much.
Are there any other questions or comments? If not, let's thank Dr. Trivedi. Our next speaker is Dr. Ruma Pal. She is the former head and professor of algology in the Department of Botany, University of Calcutta, and she is the founder member and honorary secretary of Probi Charity Research Foundation, life member of Botanical Society of Bengal, fellow of West Bengal Academy of Science and Technology, Professor S.C. Dixit Award from Indian Botanical Society. She is a recipient of that. Her main area of research is algal diversity study, morphological and molecular and ecological modeling, algae and environment, phytoremediation, carbon dioxide sequestration, algal biotechnology, nanotechnology, biofuel production, aquaculture, mass cultivation and technology transfer at rural level. Dr. Pal is going to talk about scope of algal biotechnology, application and entrepreneurship development. Dr. Pal, please.
Thank you, Professor Thakur, for introduction. And I just express my heartiest thanks to organizer, especially Professor Dhruvajyoti Chattopadhyay, our very favorite Dhruvoda, and Professor Arun Lahiri Majumdar. We also call them as Arunda. So it is my pleasure to give to share my experience on algal biotechnology. Totally, I will share today only the few technologies which are related to cyanobacteria and algae and which have already have transferred to a field level. And as time is very short, so I will just give some brief idea. কোনটা এটা তো হ্যাঁ এটা তো পয়েন্টার So as many students are there, I will give some introductory comment regarding the cyanobacteria and algae. Cyanobacteria, you know, most primitive prokaryotes. Already in my previous lecture, she expressed some views about cyanobacteria. They appeared at 3.2 billions of years ago and made the earth green. So what is the importance of cyanobacteria? There are many of my students are here, I can see. So always I used to say in my first class, because they made the earth oxygenic. And after that, the eukaryotic algae, red algae, then green algae and golden brown algae, the basic 4 types I can say, they appeared one after another by endosymbiotic process and made the earth colorful and suitable for other organisms to grow. Now what cyanobacteria? Cyanobacteria actually expose lifestyle quite similar to that of eukaryotic algae. Now in present day phylogenetic analysis, they are far apart from algae. Cyanobacteria in separate clade like bacteria and algae in separate, totally separate clade from plant system, green plant. But as they share many characters similar, both of them are thalloid, both of them show oxygenic photosynthesis and many other features in reproduction, sporulation and life cycle pattern, they are commonly called as algae. But for a true botanist, the terminology should be different for cyanobacteria and algae. Until late 70s, cyanobacteria treated as plant. I told you it was treated like plant, cyanobacteria and algae, because all of them share the photosynthetic process very similar to higher plants. Now it is in separate group. Now here also I will mention today algae including both cyanobacteria and algae, because I am going to discuss about some biotechnological implications. So algae occur wherever there is water. This is the basic character. In ponds, lakes, streams, you will see different colored mats over surface and many other places. You can see algae and cyanobacteria. In this ice covered area and in hot spring, I told you morning to Shudipto, if you can see the stress protein here, and this is Coccidiopsis, the cyanobacteria of desert, which can survive without water more than 10 years, the extremophiles, and the, you know, the seaweeds, which form the kelp forest beneath the sea. All are considered under the same nomenclature, commonly the algae. And they have lots of variations starting from unicellular, colonial to filamentous form and the thalloid plant. But this thallus is different from, it is leaf-like, but it is far away from leaf because they don't have the vascular system. It is, you see, the plant-like, the kelp forest, but they are without any vascular bundle. Now in West Bengal, there are different ecological zones. So we started 20 years back when I joined in Calcutta University, searching the strains. For biotechnological exploitation, I always believe that local strains are much more profitable and easy to cultivate. You know, for any biotechnological program, you need the biomass. Algal biotechnology is equivalent to algal biomass production. I must say, there is no complicacy, only you have to produce the biomass. Now identification and characterization, that is very tough because they share many characters similar to each other. you can see the unicellular cyanobacteria and algae similar, but when you will do for molecular characterization, you will see that they are far apart. They are not single genus. So morphotaxonomy, scanning electron microscopy, and this pigment, all cyanobacteria and algae are cascade of several pigments. So by FACS or fluorescence activated system, the pigment may be identified if there is confusion, and ultimately the molecular markers will say you. But by molecular studies, you may not reach to the species level sometimes. So you have to stay up to genus. It is better to consult more literature available regarding this. But for any biotechnological program, you need to isolation and cultivation of the biomass. So we isolated more than 300 cyanobacteria and algae from entire West Bengal, but potential genera we went for biomass cultivation, large scale cultivation, like it is Chlorococcum, Chlorella, Spirulina, and Nanochloropsis and all, many 20 or 25 for mass production. Today I will be confined to biotechnological exploitation in aquaculture, in biodiesel production, phycoremediation, and phyco-nanotechnology. In aquaculture or fish feed technology, it is very interesting but very simple. You know, fishes are growing in aquatic pond where lots of algae are there and they are fed on the phytoplankton. But if there is large scale eutrophication, then bloom comes and they kill all the fishes. So there is always a good relation, or you can say important relation, between algal growth and fish production. But these algae, when we started, and algae based fish feed production is very important, and it increases the fish productivity to a larger extent. You see, what we did, first, what are the different steps? Selection, exploitation of local algal strains, nutritional parameter analysis, mass cultivation by low cost method, algal feed preparation. This feed may be different type, total algal feed supplement, and value added feed, and feeding trial. We did for several fishes. You know, this is carp fishes and goldfishes. You see, the protein, carbohydrate, vitamins, minerals, oils, after analyzing, we just mix the algal mix 50% with the control fish or market available fish. And you see the 50%, this is the VAF. VAF means value added feed. 50% commercial feed and 50% algal mix gave good results for carp, and this is goldfish. This coloration, excessive coloration, because carotenoid is coming from the algal mix, and secondary carotenogenesis is there due to algal feed. And not only that, the food quality of fishes, the protein, carbohydrate, vitamin content, everything enhanced for this algal feed. We also applied the hatchery, prawn hatchery, at the West Bengal government. It was a West Bengal government project, prawn hatchery, and it was found that with algal feed, the prawn size is much more than their control feed was based on the animal protein. So we used these algal protein and the carotenoids, and carotenoid deposition you see in the prawn larvae, and the mortality rate is also 0 in case of using algal feed, because the vitamin carotenoid actually give the vitamin A and the immunity also. So this formulated feed, then we started screening the how to minimize the cost. So in Sundarban area, large area is actually algae are growing luxuriantly. I visited several places, 30 islands of Sundarban, where in each fish varies, a large amount of algae are growing, and the people are throwing just it. But these algae, you can utilize it properly And so we started training the fishermen. From that collection, natural catch in sustainable way, we formulated the feed which gave the better results. And we extended our work through the NGO at Sundarban, and we applied our formulated feed with their applicator. You know, the fishes, there are different types of fishes. Some fishes, they take food from the surface. Some head mouth is like this. Some fish take feed like this, and different layers. So we put the applicator at different length, different depth, and it gave good results. Now I will go about phycoremediation. Phycoremediation is very important biotechnological potential for the algae, because you know the cyanobacteria especially, they appeared in such a condition where there is metal toxicity was more. So they have the mechanism to resist the metal toxicity. So we actually try to utilize them. Now, when algae are exposed to any metal, any toxic metals, there are long-term responses. So some will be accumulate and biomagnification will be there. Changes in population pattern first, from sensitive genera to resistant one. Changes in reproductive pattern, large number of spores will be produced, and algae can contaminate the food chain also through them, because there is biomagnification. And short-term response, active accumulation, surface adsorption, enzymatic changes, extrusion. Resistant algae sometimes accumulate metal in non-toxic form, or sometimes they accumulate and extrude out. So extrusion process is in bacteria also. Polyphosphate binding, the metal chelators, biotransformation, methylation, or specific gene action, phytogelatin production in exposure to metal. So we did work in one village at North 24 Parganas. This is Kalsur, and it was arsenic contaminated entire village. It is actually geological form, and the people are suffering from the arsenicosis there. And it is at least 15 years back I did this work. And then we started generally, you know, this student may know that this arsenic actually stay in different form, oxidative status. Arsenite is the more toxic one, and arsenate is less toxic. Some algae accumulate arsenite and they can transfer to arsenate or methyl arsenic, that is also non-toxic form. And this biotransformation, methyl arsenic to dimethyl arsenic acid, trimethyl, there are several products, and the toxicity level of these arsenic sugars are different. So resistant strains, what they do, they either chelate the arsenic within the cell, or they accumulate and arsenate and reduce to arsenite and extrude out. In this way, the resistant ones stay there, and some stress enzymes they accumulate within their cell. But sensitive one, they become dead and the population is changed in the contamination area. So we surveyed and collected a large number of arsenic tolerant strains. See those mainly are there cyanobacteria with large number of sporulation was there. I told you reproductive patterns, if the soil is contaminated, then they produce large number of spores, and some diatoms and euglena are recorded from the contaminated area. Then we compared the in vitro and in vivo study. In the area in vitro, there was upregulation of stress enzymes and other enzymes which are giving them the resistance. And in vitro study also, catalase activity was high and lipid peroxidation was low in vivo. And then you have to determine the kinetics, absorption kinetics, how the algae and the metal are working. Whether if it is active absorption, they can actually accumulate toxic metal within the cell, then amount will be less. And here is from these kinetics, Langmuir isotherm models, if you get the straight line, then you will be understand that the metal is on the surface. Because if it is surface, then this growth kinetics will show the straight line, because it is known from Langmuir isotherm model, the one round surface absorbs most metals, and there will be these kinetics. And we also detected the arsenic reductase enzyme in the contaminated algae from the Formidium laminosum from the contaminated site. So our aim was to design a biofilter. So what we did, we collected some Formidium from Sundarban area, which is not contaminated, and we found that in Sundarban area, the Formidium tenui, they absorb the arsenic on their surface, and you can remove that arsenic with chelators. But that Formidium laminosum, which was in the arsenic contaminated site, they had showed mostly the active accumulation. So we used this Formidium tenui from Sundarban area to design the biofilter. So we trained the villagers. It was a social project from West Bengal DST. So we trained the villagers how to cultivate this Formidium in this Kolsur Gram Panchayat, and they made the filter, and they were successful. This was patented, this filter, and it was awarded best in Algal Biotechnological Symposium at Australia. And it was patented from West Bengal government, and they did this video also, arsenic free drinking water. Interesting point is that this year in G20 summit, this filter was showcased at New Delhi last year. Now with this capacity of cyanobacteria, we started doing work at Bantala leather complex. You know, this Bantala leather complex, there is lots of wastewater is coming, though they are very conscious about the bioremediation or the chemical reduction of the sewage, this wastewater. And there, you know, we saw that the chrome water, that is very important, chrome water and the wastewater. Chrome water is high amount of chromium water is needed for hiding the skin of the animal. And the entire wastewater is highly polluted, organically polluted. So 3 algal strain, Anabina, Leptolingia and Arthrospira, were used for this wastewater treatment, phycoremediation part, and these algae were also used, the cyanobacteria, for chromium removal. In one hand, it removed the chromium from the chrome water, and together with that, it transformed the chromium to nanochromium. So it was actually it was a point from waste to wealth, and it is published in good journals. And not only that, we also did the eco-monitoring. Eco-monitoring, this, though the Bantala leather complex, they are doing their wastewater treatment plant, but the wastewater is coming out, treated water is coming out and mixing with the Vidyadhari river of Sundarban connecting canal, and we did some ecological modeling of the entry point upstream and downstream. And I am not going to show the diversity, but it was found that there was change in population from sensitive to resistant and intensity, algal growth intensity. So another project was there, our algal biodiesel project, and you know, 10-15 years there was a craze, and we got invitation from CSIR to propose a project. So we did that, you know, algal biodiesel means for just like any plants or crops, biodiesel will be extracted. But main thing was that whether there is any suitable marine strain, because principle was there, as you know, 3 parts is C. So if we can get any marine strain, then biodiesel production or any production will be less costly, and not only that, the crop plant won't be affected. For that reason, the searching was there. So we did so many algae from Sundarban. We did total lipid content, mono and unsaturated fatty acid with confocal microscopy and all. So mono and disaturated fatty acid were there. We prepared the biodiesel from chlorella and this chlorococcum. It was in the Indian standard diesel, it is at par, and he is actually DG, CSR DG at that time. We ran the B20, this microalgal biodiesel, it was CSR Nimitli project, and the then petroleum minister was in the car. But production was there, but only problem is that the cost of the biodiesel was high. Because biodiesel, you know, it is high value, low cost product. So now government is going on to give the projects on not on biodiesel but biofuel, different type, and algal oil. In that case, I feel that high value, low volume product will be best for algae. Like algal oil, not biodiesel, algal oil for diatoms and all, it is very good. So now it is, I am in DBT for this monitoring committee, but search is going on. Now I will end with this, our nanotechnology. We are working with algal nanometal formation and other. So these are the nanometals what we prepared in our laboratory based on different cyanobacteria and algae, both gold, silica, nano gold, nano silver, nano iron, nano chromium, silver silicon composite, copper silicon nano composite, silver silicon, nano dendrite, that these stem-like structures, gold, silver, nano alloy, etc. It is very simple process. This is phycosynthesis of gold nanoparticle. You see the biomass when exposed to gold chloride solution, it turns red. Initially it was very difficult to identify what was that, because we were working with precious metal separation. Then we came to know that this is actually nano gold. So these are the microscopic image of the algal thallus producing nano gold. You know, in ancient time, the Egyptian used to use this nano gold for painting purpose. So it is just like painted cyanobacteria and algae with pink nano gold. These are the extracted nano gold from algal, and this is actually some fluorescence property of the nano gold particle. You know, these are chlorophyll control, but when this nano gold or nano silver are produced, then their fluorescence property changed. These are green, these are actually the carotenoid. Much more synthesis of carotenoid and then nano gold. So in this scanning electron microscopy, you see the control one, when they produce nano gold, the large amount of polysaccharides are synthesized. Here the polysaccharide part of the algae are the actually bioreagent acting as. But here you Anabina spherica, this is, you see polysaccharidic sheath is there, but when exposed to gold solution, the polysaccharide disappeared during formation of the nano gold. That means these polysaccharides are used as bioreagent. These are transmission electron microscopic figure. This is of Lingbia. You see the nano gold deposition within the cell and outside the cell in polysaccharidic sheath. So lots of paper at that time, 2006, 7, 8, 9, 10, lots of paper, and it was exciting. But after that, we felt that people started asking the journal, from journal, what will be the fate of this nanoparticle? Because for any nanobiotechnological purpose, you need monodisperse, one shape and size. But in case of algae, all are polydispersed. Some are circular, some are hexagonal, some are triangular like this. So we started thinking what to do. We started separating different parts of algal thallus. This carotenoid, protein, polysaccharide, but everywhere polydispersity appeared. And this is, this isolated chloroplast and isolated thylakoids also. So all showed the polydispersity. Then we actually changed the pH, and we found the pH of the solution, reaction solution, that is the driving force. And this pH 5 only rod shaped nano gold prepared, and this is only circular at pH 7 and 9, and pH 9 all are similar sized nanoparticle we obtained, gold nanoparticle. Then we went for mass cultivation of nano gold particle in extracellular condition. In large tank, those algae which can produce extracellular nanoparticle, we produce huge one. This is silver nanoparticle, same procedure, but after nanoparticle production, actually you have to test with UVVs, DLS, zeta, FTIR, XRD, TEM, SEM, AFM. And silver nanoparticles, they have antibacterial, algicidal, anti-cancerous activity, like you can produce silver nanoparticle by any way, but one achievement we had to get it was the joint work with Professor Anjan Dasgupta of biochemistry. He said, I worked a lot with him, collaboration with this nano technology. He said that as this is a reduction process, it may be that it is forming the anoxygenic environment. When algae or cyanobacteria is producing silver to nanosilver. So he designed a device, and he said it may be effective for any purpose like food preservation. So he prepared a device where the algae are growing, and algae and silver nitrate solution, and on nets the grapes were, initially it was done in beaker, and the results were very fantastic. Then when the nanosilver production, it can, fresh fruit you can get after 1 month in room temperature. So we got these patents, both Indian patents and US patents, and this is the device, fruit preservation device based on the nanotechnology. And diatom nanobiotechnology is also important because botany students, diatoms are actually enseeded with frustules, and that diatom frustules are totally nanosilica. So if you cultivate diatoms only, you can get a large number of nanosilica, what you can get in diatomaceous earth also. Diatomaceous earth is produced from fossilized diatom. So we actually isolated this diatom amphora from Chilka, and we culture it, and you see the diatom frustule, it is ornamented, and so it is designed also for identification it is needed. And this with computer science section of our Calcutta University, Dr. Sanatan Chattopadhyay, we designed one nano chip from the diatom frustule. This nanosilica is used for production of nano chip. It was actually from diatom frustule, this nano chip. And this nano chip was characterized, optical characterization, physical and crystallographic characterization, and it was found that it is good for further use. And that I already told, diatoms are a very good bioreagent for producing the nanodendrite, these stem-like structures, and these nanodendrites are very good for drug delivery designing. Then nanoiron also produced nicely from algae, and these nanoiron, they have tremendous implication in this chromium removal, pollution removal. So one principle we use that rather than isolating nanoiron from the algae, it is better to use the nanoiron impregnated algal biomass. So this reagent will be very easy to handle. So we used that leptolingbia with nano iron for chromium removal, and it gave good results. You see that it came in algal research and this Journal of Phycology. The nano iron particles, they are all uniform and inside the cell, and this is extracted one. So another work from this algal nano iron impregnated algal biomass we started very recently. We proposed nano phyco fertilizer. You know, algal biofertilizer, it is very known. So we used ulva powder for nano iron production, and we gave all to rice plant. The only DAP, this is the plant for DAP, and the productivity, you see the productivity grade, increase in grade, and this is only algae, and this is nano iron isolated, and both algae and nano iron. And these are the algal source and produce, and this after characterization, nano phycophyte, and it is applied to field. In Medinipur field, you see the farmers are using, and these are the grains size, and quantity and quality wise also is maximum iron content in this nano phyco fertilizer. And all these analyzed the plant quality, it was better. And interestingly, post-harvest practice, after the rice has been taken out, the keshari, the khashari dal, the lathyras, they used to cultivate in the land for this nitrogen, increase in nitrogen. And that plant has also been increased. Their growth is also more due to where nano phyco fertilizer was applied. And now that work actually with this rice work, we collaborated with Dr. Soumitra Pal of our department, this nano phyco fertilizer. And now we are applying this nano phyco fertilizer to the medicinal plants. We have just started with Professor Shantanu Pal of our department. It also gave good results, just growth in this nano phyco fertilizer and control. And this work is going on already. The rice work, our student is received the best paper award in World Anthropology Congress, in the Sustainable Agriculture, and in this Bangla Bigyan Congress in Kolkata chapter. So for implication, we actually algae hub at Basanti Sundarban, just to training the rural people. We used to give regular training, and they are producing now spirulina. This is a spirulina project. The school boys of Sundarban, they are producing spirulina at Sundarban. The production was going on
Dr.
3-4 years back. Yeah, I have completed. And these the rural people are producing food product, biofertilizer. And in our Calcutta University campus, we established this algae cultivation and culture collection and cultivation unit, and one algae herbarium for the students who can visit. Thank you. These are my scholars working on different fields.
Any comment or question from the student? Yeah.
Students ask first. I will ask. Okay, it's very nice and exciting talk, I think spanning 40 years. So you have shown a lot of applications. So I'm just wondering whether the cultures are preserved somewhere, or how, like every time you do something like.
No, in our culture collection center, we are maintaining. And we are maintaining, sometimes culture comes bad, you know, because we don't have much manpower. So scholars are, then we used to go collect and isolate and this biomass production and all. It is going on. But though I am retired last year, till date they are taking in charge, some few students are working of some other professor. After that, I don't know what will be the fate of this in this center.
And when I see you teach people to grow them in these large bottles, are they sterile or how do they maintain contamination?
No, there are two types. If you want to maintain sterile, then you make the media sterile and do in front of laminar airflow and like this, then it will become sterile. And during isolation, in this agar plate, you put on this antibiotic. So the antibiotic resistant strain will come. Clear? So it will be bacteria free. And for algal culture maintenance, you have to maintain the log phase. Because we can't give mercury chloride or anything, sir, what you showed, these ciliates are moving, you can't. But if you go on this subculturing rapidly with this algal media, little bit, this after washing with betadine and add little bit antibiotic, then it will grow.
Okay.
That will be sterile. And but if you do this phycoremediation work or fish feed work, then we go for open pond. There may be infection, but if you maintain the log phase, then majority, 90% of the population of your own strain. Clear? And biofertilizer, if there is any contamination, there will be no harm. It will be algal consortium mainly because there is no carbon source. Some bacteria may come because there is polysaccharidic infestment on the algal surface. But you keep on maintaining the log phase. Then it will be minimized.
Ruma, my apologies for including the cyanobacterial sessions in the plant biology genomics workshop. Because you said the cyanobacteria are very different, they shouldn't be, next time it won't happen. Secondly, is there any gender bias in algal research?
No.
See, out of the 15 workers that you worked with, only 4 are men.
No, no, sir, I didn't show all. But I like a girl student more.
Yeah, oh, okay.
It is
that's a very frank confession. Thank you very much.
Because I can manage them very well. Or boys, girls prefer me, it may be.
Okay, okay.
Is there any other question or comment? Please go ahead. Make it brief. We are running late.
Good evening, ma'am. Ma'am, in heavy metal filtration, the picture which you showed, wherever we are growing a culture, there might be some death. And as I have seen in my lab, whenever these cyanobacteria die, there are some other microorganisms that are festering on it. So, in case, how to prevent that?
What I told you, that you maintain the log phase. 14 days, 14 to 21 days of inoculation is the log phase of the culture. If you inoculate and keep for 2-3 months, then excessive polysaccharides will grow and bacteria will come. So you keep on subculturing after 2 to 3 weeks. And you isolate in the agar plates with antibiotic and wash the biomass what you get. I told you I prefer always local strain. I didn't buy a single strain from any culture collection. We collect, then first you wash with betadine and keep it in running water for 2 to 3 hours. Then go for the streaking method with little bit antibiotic and then it will grow. And then from there you grow in another series of inoculation.
Ma'am, in the final stage, when we are growing in a mass culture, then sterilization becomes a problem because big jars or containers cannot be sterilized. In such case, if certain other microorganisms like bacteria or fungus may come in, how to prevent that?
No, actually in large scale, it closed photobioreactor for any precise chemical production, just you want to produce beta carotene. Then you need the strain, pure strain. So in that case, entire system should be sterilized. Passing hot water, boiling water or whatever, you have to sterilize and keep it close with the carbon dioxide flow and all. But for other phycoremediation and biofertilizer, you go for open, because biomass cost is the most important. Clear?
Okay, ma'am, thank you.
And for biofiltration, you grow, I saw in Atrey's lab also, they are growing in sponge and all. You grow in different media, glass wool and all. The cyanobacteria will grow. Ultimately, the filter will be produced. Okay?
Thank you, ma'am.
Okay, sorry, we have to come to a close because we have a valedictory session right after this. So, thank you, Professor Pal, and thank all those who are present. Okay. With
Thank
that,
you.
this technical session ends. Right.
Thank you, sir. Sir, now may I request Professor Thakur to kindly felicitate Professor Ruma Pal and Professor Sucheta Tripathi, ma'am. Thank you, sir. Thank you, ma'am. Now, may I request Professor Arun Lahiri Majumdar, sir, to kindly felicitate Professor Ashok Ranjan Thakur, sir. So now we will have a short tea break, after which we will have the valedictory session where the best poster awardees will be revealed. The students and the participants can go to the third floor and for the tea. Yeah, it will be for 15 minutes. The tea break will be for 15 minutes. And may I request our distinguished guests to be seated here. Kindly be here. And the students and the participants can proceed to the third floor.